Yeah, lovely. Well, it's an absolute pleasure to be here. I attended the Newcastle participant event and got some really difficult questions, so they've set the bar high. As Mark said, I'm Ben Lacey. I'm an epidemiologist and public health physician by training, and I lead the epidemiology group at the University of Oxford. So we provide the scientific support to Naomi Allen, who's the chief scientist, and others.
Excuse me, can you go a bit louder, please?
Go a bit louder? Is that better?
Yes.
Great. Yeah, so we provide the kind of scientific support for Naomi Allen, who's the chief scientist, and others in the UK Biobank. And I'm going to give you just 5 minutes, really, on the story of UK Biobank so far, so kind of very brief overview. So as most of you are kind of probably aware, the kind of key overriding aim of the study is to better identify and understand the causes of disease. So that's the genetic, lifestyle, physiological, environmental determinants of disease, with the key aim of leading to new and better treatments and preventative measures. I mean, that's the reason that we're doing it. And there's been lots of studies that are kind of similar to UK Biobank before. So just before UK Biobank was kind of set up, there were kind of other studies that took groups of people and followed them over the time, so very similar to UK Biobank. But they all tended to be either very small with very detailed information, or they tended to be very large with relatively limited amount of information. And the really key insight, the thing that makes UK Biobank unique, is bringing those two together. So at scale, where we've got 500,000 participants, it's that kind of depth, it's the kind of capacity to really understand disease from the information we have, and now the length of follow-up. So we've got over kind of 15 years of follow-up, so it really is unique in the world. So recruitment, as you remember, was between 2006 and 2010. Participants were registered with the NHS, and they all lived within about 25 miles of the 22 assessment centres. So there's 44,000 who are from Leeds, and slightly less from Sheffield, but I understand there isn't too much of a rivalry there. So this is one of the assessment centres that all of our participants, I'm sure, will kind of recognise. There was a touchscreen questionnaire, that was followed by a verbal interview, and then physical measures and sample collection. And those biological samples continue to be stored in these large warehouses with our kind of robotic arm that kind of collects all those samples efficiently. So these are the characteristics of all the participants in the study. The average age was just over 50 years, just over half were female. Most were of white ethnicity, but still just under 30,000 from other ethnicities. And they came from a very wide range of socio-economic backgrounds, some ranging from very affluent to more deprived. And then the participants have followed up through linkage with NHS datasets. So that's death registries, cancer registries, hospitalisations, GP information about half of our participants, and then during the COVID pandemic, linkage with the national data sets that were very helpful to researchers. But one of the key things about UK Biobank is rather than just passively follow up participants, the data that we have, the information that our participants have provided is being continually enriched. So straight after that initial baseline assessment, there was a resurvey in 20,000. Many of you would have completed our web-based questionnaires, and thank you very much for that. 100,000 participated in our physical activity monitoring, which involved wearing this wrist-worn accelerometer for 7 days. And then the biological samples that you've been produced, we've been kind of busy in the background analysing all of those so that they can then turn that into data that researchers can use. And then lastly, our multimodal imaging. So that's, we're targeting 100,000 participants. And this is a really big undertaking. So previous large-scale studies of imaging were a matter of a few hundred, maybe a few thousand. So the idea that you could get this in 100,000 participants is really game-changing. So who's using this study? Well, just a few years ago, it was predominantly the UK, with some from North America and elsewhere. It's now a truly international set of researchers. So it's gone from about 50-50 new registrations in 2016 to more like 80% from international research centres and 20% from the UK in 2022. There's now over 30,000 researchers from around the world working on this UK study, and just under 5,000 scientific papers. So I thought I'd show you this kind of word map of just the types of things that the researchers are working on. So you can see genetics is big here. A lot of genetic data has been released recently. That's kind of been a real focus of researchers' work. There's cardiovascular disease, cancer, as you might expect, some on cognition, imaging. But also there's lots of other kind of research focus here. So oral health, pain, sleep, ethnicity, aging. And it's really, that's that breadth. It's that breadth of data in the UK Biobank that's its strength. So it's the size together with the breadth. So as we kind of we get more information on imaging, that's being added to this wealth of other data. So researchers interested in dementia, they don't just kind of look at a kind of limited set of lifestyle factors. They've got genetics, physical measures, physical activity, the online surveys. So it's that combination, size, depth, and imaging that's really kind of driving researchers to use this study. And lastly, just a big thank you to all of you, the UK Biobank research team, and of course our government and charity funders. Thank you.
It's wonderful to be here, and my goodness, standing room only today. It's fantastic. I'm a neurologist, as well as someone interested in imaging and UK Biobank. So one of my major areas of interest is in dementia. And it's not in studying dementia per se, but it's in figuring out how we can avoid it. Those of us who are getting a little bit older worry about it more and more with every day. UK Biobank offers a really globally unique tool for beginning to understand something about how to identify what precedes dementia and how to stop it. You've heard a lot in the last few minutes to remind you about the size, the depth, the number of investigations that all of you and others have contributed to, the length of time of follow-up, which is critical for understanding a disease, because we can look and see the way people are now and then what happens to them in the future, and that's really absolutely fundamental to understanding what the factors are that determine disease. And then finally, you just heard how many people all around the world are using this data set. It was really gratifying to go to Africa last year and see how many people were using the data set, and they had so few other resources to do science, but UK Biobank was helping them to help everyone understand disease better. Now, I said, hey, we can watch disease appear, and just to make this really graphic, and I'll focus on my own areas of interest, which are down at the bottom here, disorders like depression, Parkinson's disease, Alzheimer's disease. There are people with all of these diseases who are represented in this large population now, but what is more important is, in a few years, there are going to be many more. So there are 5,000 people, roughly speaking, with Alzheimer's disease who were in the study in about 2020. By 2027, unfortunately, about 17,000 people will have developed dementia. And that means that about 12,000 people between 2020 and 2027 don't yet have it, but are starting to have the early signs. And if we can study those now through this resource, we can begin to understand what the factors are and how we can intervene to help others. Now, you won't have seen this, but you'll certainly have seen news reports of all of the risk factors that are associated with dementia. And in fact, there were reports in the newspaper today about, for example, healthy lifestyle, which is one of the factors here. We've heard about depression, smoking, social isolation, physical inactivity, and all of these factors. These are all risk factors for Alzheimer's disease. But what a risk factor means is that you see the risk and you see the disease later, and they seem to be associated. It does not mean that they cause it. And the example here is, this is a cartoon, of course, but this man observes that everybody who went to the moon has eaten chicken. And his friend says, good grief, chicken makes you go to the moon. Now, of course, that's absolutely wrong. It's an association, but not a cause. And the size and the genetic data that's associated with UK Biobank actually allows us to tease these apart. So, for example, here, looking at the American Heart Association's Life Simple 7, the things that they suggest are the modifiable risk factors for late life dementia, like blood pressure, high lipids in the blood, control of blood sugar, smoking, weight, diet, activity. One can see that all of these are associated. This graph shows, for example, that blood pressure, this is a systolic, the high blood pressure end of 200. This is the relative risk, and it shows that once one gets up into the high hundreds, the risk of Alzheimer's of dementia rapidly increases. But the question is, which of those factors actually cause Alzheimer's disease? And one can begin to pull these apart using genetics, because we can figure out with studies like Biobank, what the genes are that cause high blood pressure, for example. We can see what the genes are that cause Alzheimer's disease, and then by using studies like UK Biobank, we can apply sophisticated statistical tools known as Mendelian randomization to look for evidence that one causes the other. And here, this shows very robustly that systolic blood pressure, the high end of blood pressure that you get measured in the doctor's office, clearly is causally associated with Alzheimer's disease. Why is that important? It means that if you reduce your blood pressure, that is one of the factors that is most likely to reduce your risk of subsequent Alzheimer's disease. Now, let me give you another example. I was talking to you at the very beginning about seeing disease before it happens. Here's an extraordinary example that was done by Cynthia Santor and her colleagues in Cardiff, where they looked in the UK Biobank population, and I won't go through this in detail because the print is too small even for me to read from here. But the point is that some of you may remember that about a decade ago, people were asked to wear an actigraph watch in UK Biobank, and they wore it for about a week and collected movement data. Now, it turns out that when one looks at the movement data, a specific type, the acceleration in the step, in two groups of people, that group that went on to develop Parkinson's disease within a 7-year window of when they wore the actigraph watch, and that group that went through that same 7 years in UK Biobank and were matched for age and sex, and they didn't develop Parkinson's disease. You can see a clear difference. The acceleration in the step, the quickness with which they move their foot, was very different between the two. And it turns out, I won't go through this long table, but if one looks here at Parkinson's disease, there's a very clear difference between the healthy people in UK Biobank, those people who have never developed Parkinson's disease or another movement disorder, and those people who develop Parkinson's disease, which you see here. And it's very specific for Parkinson's disease. You don't find it with osteoarthritis or depression or other disorders. So that's seeing disease before it happens. Now, the imaging we've heard referred to is something that I'm very excited about with my colleagues, because it allows us to look directly inside the body. And when we can look inside the body, we can develop what we call imaging endophenotypes. And that's just a fancy word for a signature on the brain images or the body images of a future disease. And that's also something, because the imaging that's used in UK Biobank is very similar to that that's used in the clinic. With these discoveries, we can translate them to the clinic. It's particularly important for dementia, because if we look at, you'll hear more about this from James Cole in a moment, this is a study of the change in brain shape and volume in different regions with age. This is age along here. These are our youngsters down here in their 20s, showing how the brain is developing. But all of us who are over 60 or so, unfortunately, we're on the downward swing. And the extent to which where we position ourselves there tells us a great deal about how genes, environment, and lifestyle are affecting our brains. And one can analyze these brains on an incredible scale. So you all see this. This looks like a nice brain image, more or less, doesn't it? But in fact, that is an image that's a composite of the first 5,000 brains that were studied. And the quality of the data that many of you are providing is of such high standard that we can mathematically warp the brains into a common shape and study them all together, which allows us to really look at the associations between brain size and shape and all of the many things that we see that people have contributed through questionnaires or through their medical records. And particularly I want to emphasize that lifestyle factors, which are a huge range of about 5,000 variables, lifestyle and comorbid diseases, the other things that one has, all have a big impact. And you can see that by these little dots here, which represent the degree to which they vary from the normal in the population, a huge impact on the way the brain appears. And you'll hear, as I say, much more about that from James. But the point is, the brain is a remarkably sensitive organ to our body health. And in UK Biobank, moreover, the kinds of imaging, while relatively straightforward and very close to what's done in the clinic, allow one to elaborate on this using more sophisticated approaches analytically to discover very new things. Carla Miller and her group have looked at the images of the brain and teased out a way of imaging the distribution of iron in cells. And that's really important because iron in cells in the brain is associated with dementia, and she was able to link this through the genetics data that is provided in UK Biobank to the genes that are responsible for it, identifying in particular a gene known as the hemochromatosis gene, which we used to think was only important in a rare disease of children, but it turns out that this gene also is linked to neurodegeneration. It affects the way the brain handles cholesterol and other lipids. And here you see, if you mutate a mouse, so it expresses that rare variant found in the human population. You can't really see this very well, but this white area is the water part of the brain. And here in the mouse with the mutant form of the gene in it, that becomes very big, indicating that the brain is degenerating. And the final example I want to give you is a real life one. This is moving from mice back to all of us. And during the COVID crisis, we had just imaged a large number of people before the pandemic came. And then many people all across the country, perhaps some of you, were generous enough to come back for a repeat imaging visit after you've been tested for SARS-CoV-2 serology. And you came back in the middle of the pandemic, and people looked at your brain, and it's really extraordinary. Despite the fact that these were largely mild infections, we discovered evidence, and this was led by Steve Smith and his group in Oxford, for a relative loss of brain volume in the base of the brain where the nerve from the nose, the olfactory nerve, connects. And moreover, the extent of this change in volume, which did not occur in people who had not did not show serological evidence for exposure to SARS-CoV-2, which you see across the top. It showed that with increasing age, the amount of brain volume loss was greater. So we think it's really important. And finally, I'll just highlight, we're always trying to think of new ways of adding to the resource. Some years ago, there was optical coherence tomography, a little eye scan you probably had when you went to your optometrist. Adding that in looks really exciting, and it's going to be done with the new re-imaging. In fact, it turns out that measurements of the nerves in the back of the eye, you can't read these numbers, but suffice it to say here that people with progressively more impaired cognition as they age, this is measured on a single test, the number of incorrect repeat scores, as the nerve fibers at the back of the eye become thinner, going in this direction, the numbers of worse scores increase. So we think it may provide a very cheap, very fast window into the same processes of neurodegeneration that we talked about with brain scans. And finally, I want to close with another thanks to everyone who's been involved with Biobank. It's a huge privilege to be contributing to it, even in small ways. It's been a remarkable partnership between the government, between companies, and of course between all of you and your colleagues across the UK. So thank you very much.
Well, it's a real pleasure to be here. It's great to be involved in this. My parents are participants in the UK Biobank, and I was jealous when I was telling them that because they hadn't got invited to this session, but hopefully there'll be one in the East Midlands where I grew up at some point. So I'm going to talk to you today about my research measuring the brain's biological age, or as I like to call it, how old is your brain? And first of all, we kind of ask, why do we want to know how old our brain is? Well, we know for a fact that our brain changes as we get older. The structure of the brain macroscopically changes. Post-mortem, the brain of an older person will be smaller and lighter than someone who dies when they're younger. However, it's good news that we don't need to just look at post-mortem, and we can use these lovely MRI scanners to have a look at the brains as we change while people are still alive. And Paul already touched on some of the evidence that shows that the brain is indeed changing a lot as we get older. And I had the privilege of being involved in another study which is very different to the UK Biobank, but does actually have more than 100,000 MRI scans in it. This was a gargantuan effort by collaborators in Cambridge who actually acquired, somebody mentioned earlier that the original older studies of the brain and neuroimaging are much smaller. What they did was get together more than 100 different separate studies and compile them into one huge composite database. And this is across the lifespan. I wouldn't expect you to read these, but there are lots in there. If they added the UK Biobank by itself, it would probably be about the same size when it's finished as all of those put together. But what we can see from this study is that indeed the brain does change as we get older. And this applies to the grey matter of the brain, the white matter of the brain, the subcortical regions all get smaller, and the ventricles, which are the spaces inside the brain, get larger as we age. So we know the brain is changing in structure. We also know the brain's function is changing as we get older. So this is a classic slide that basically shows that we experience something called cognitive aging. And cognitive aging is essentially the slow, steady, generally very moderate decline in the majority of cognitive functions that happens as we age. So things like memory, information processing speed, executive function, all of these will change gradually as we get older. There are some things that stay maintained, like world knowledge and vocabulary. They tend to stay quite consistent across the lifespan, but generally a lot of things do change. Some of you may have heard of this. There's a condition that's referred to as mild cognitive impairment, which is often associated with increased cognitive aging, so more cognitive aging than you would expect. And people who have mild cognitive impairment have an 8 to 10 increased risk of actually progressing onto dementia. Not everybody who has mild cognitive impairment will get dementia, certainly, but the risk is higher if you do. So we know that brain aging and brain changes associated with aging are universal, but they are not uniform. So they do affect everybody differently. And you can see this. This is some results on the prevalence, or the incidence, I should say, of mild cognitive impairment, and that changes with age and becomes increasingly higher. But what we know is that there's a lot of people who have MCI, who don't have MCI. There's a lot of people who have MCI who don't get dementia. and that your age of onset for these things is different. So clearly everybody's brains are aging in a different way. So the data I've showed you already is just about people's brain structure, but we can use MRI scanners to look at the brain in lots of different ways, and Paul's already mentioned a few of these, but I'm just going to go over this very quickly. These are some of the different types of MRI scan that you can get, and the sorts of information that you can apply and have been applied to studies of the brain's aging. So we can use T1 weighted MRI to look at how thick the cortex, the outer surface of the brain is. We can use T2 flare MRI to look for lesions or damage to the white matter structure of the brain. We can use something called arterial spin labeling, which looks at the blood flow. We can use magnetic resonance spectroscopy to look at various neurochemicals in the brain. We can use diffusion-weighted imaging to look at the structural connectivity of the nerve fibres in the brain to see how well connected they are to each other. And we can also use functional MRI, which looks at oxygenated blood flow around the brain to see how that relates to patterns of activity, brain activity. All of these things have been shown to change with aging. So we really can leverage lots of information about the brain just from a simple MRI scan. But what my research wants to do is not just understand how the brain is changing as we get older, but to actually predict how old somebody is based on their MRI scan. And this is how we do that. So normally we start with a very large data set of people who we have brain scans of and we know their chronological age. We then build a statistical model, which we call a regression, where it takes their brain imaging data and it predicts how old they are. And then we validate that. So we take out part of the data set that we've used, and then we train our model to predict age from neuroimaging data in part of it, and then we make predictions in the bit that we left out. And we do that iteratively until we have predictions on everybody to know that, see whether our model actually works. Can we predict somebody's chronological age? If we're happy with our model and the coefficients that we've learned, we can then take new data, people whose age we don't know, and we can run their brains through this statistical model that we've built, and we can get a predicted age. And then finally, we can take their predicted age and we can compare it with their real age. The idea being that if you are of normal health, your brain age should be the same as your chronological age. If you are healthier than average for your age, you would have a younger appearing brain. And if your brain is less healthy than would be normally anticipated for your age, you would have an older brain age. And we've used this paradigm in lots of different contexts, but we were lucky enough to use it in the UK Biobank. So this is a study of 6 of those different neuroimaging modalities that I was just talking about. And this was the first study to combine together 6 different things to predict chronological age. And on this scatter plot here, you can see on the axis here at the bottom, we've got age. This is the real chronological age of people. And then here we've got what I refer to as the brain predicted age. And you can see that, broadly speaking, we're doing quite a good job of getting a perfect prediction would be this dashed line here, which is where age and predicted age are the same, but more or less we're getting a scatter of points in and around the predicted age, the real age of the person. So on average, the error, so the difference, the mistake that the models make, is about 3.2 years. And given that the participants in the UK Biobank range anywhere from early 40s up to their 80s, we think 3.2 is actually quite good performance for this. And yeah, as I said, this is the first time that we've used 6 different types of MRI together in one model to predict how old people are. This was a study, I should have said, this was a study in one of the first releases of the UK Biobank imaging data in about 20,000 people. So we ran this model and we took predictions of people's age and we looked to see whether their brains appeared older or younger than would be expected. And then we looked to see what the difference between people's age and their predicted age was related to in some of these other things that we've measured in people. And we were able to show that people with older appearing brains, older appearing brains were associated with high blood pressure, with the prevalence of stroke, with prevalence of type 2 diabetes, with smoking, with alcohol intake, and even some measures of cognitive performance. So clearly, having an older appearing brain is actually associated with some of the things that we know are also risk factors for dementia. This was just a cross-sectional study, and we are planning to follow it up. But we do have some interesting evidence from a different study that didn't use Biobank, where we had, this is a study done at King's College London, where we had just over 1,000 people from a memory clinic who'd undergone MRI scanning, and their brain scans were linked to their electronic health records, and we could see who had gone on to get dementia out of the people that attended that memory clinic, out of those 1,100 people. And it turned out that having an older appearing brain was associated with progression to dementia. And that was, some of these people were followed up to 8 years after their brain scan. So it does certainly show that our brain age measure is sensitive to some of the things that also increase your risk of getting dementia. And in due course, we will hope to apply this exact same study design to the data from the UK Biobank when more time has elapsed and the sample size has increased. So, in summary, we can measure, or at least I think anyway, we can measure the brain's biological age using MRI. Different types of MRI can be combined to help make those measurements better. Lifestyle and health factors are associated with increasing one's brain age, and that relates to your risk of getting dementia. And I think how old your brain is, it does matter. So thanks for your time, and I'm happy to take any questions.
Thanks very much indeed, folks, for having me. I'm delighted to be able to come and speak to you all about the fabulous resource that you've created. I mean, it's a huge pleasure to work on the data and to meet a few of you in person too. So I'm interested in exploring brain and cognitive aging pre-dementia. So what does that mean? Well, so James, who was up here earlier, did a very good job of prepping you all for what that means. So unfortunately, depending on who you read and which data sets you look at, from about 20 years old onwards, many of our cognitive skills are on the downwards trajectory, on average across the population. But there's lots of variability within that. So it's not an inevitability that you're going to steeply age in your thinking skills. But on average, one can expect to do that to a degree. But importantly, there are several things that we're looking at that are trying to understand why it is that people differ in their brain and their cognitive aging trajectories. What I thought I would do to begin with, though, is show you a little bit about brain aging, and again, you've had a good bit of priming for this in the previous talks. And so what I've done here, I hope everybody can see the red dot, but I've arranged brains here. So this is just looking face on, a cross-section of our brains. This is the skull on the outside, and I've arranged them from top left to bottom right in terms of the amount of atrophy, so overall shrinkage, if you like, that one might expect to see with aging. And you can see, if you compare, for example, this top left brain to this bottom right one, you can see how the outer cortex, where the grey matter sits, where most of our brain cells are, has shrunk away from the intracranial vault inside of the skull, and how also the ventricles, where our cerebrospinal fluid sits, have yawned massively. So the brain is shrinking from both the outside in and from the inside out. Don't worry, there is good news coming, don't worry. And so this is a measure of atrophy, so how much our brain is shrinking relative to how much we used to have. A different type of scan now. So this is now looking down, if you like, on top of people's brains. And again, same sort of thing. So from top left to bottom right now, we've got these white matter lesions, or white matter hyperintensity. So these are the white matter is the connective tissue that allows information to be efficiently transferred between the distal sites of the outer grey matter cortex. And this is lesions, damage to the white matter, increasing from top left to bottom right. The interesting thing about these scans, they're all different people. They're all 73 years old. So it shows you a couple of important things. First of all, as I said before, it's not an absolute inevitability that you're going to experience a large degree of brain aging. I mean, as I say, at 73 years old, some of these individuals' brains wouldn't look out of place among 40-year-olds. But it also shows the variability with which people who are non-clinical, they're not in a clinical population, not demented, experience these different hallmarks. And it raises the question, how is it that you arrive at older age with a brain looking like that, and how do you avoid having a brain that looks like that? And so this is John Scott from one of the Edinburgh Lothian birth cohort studies, and a 3D print of his brain motivates this question, doesn't it? Why do some people age more in their brains and their cognitive skills than others? And that's the fundamental driving question of a lot of the research that we do at Edinburgh. Now, although, as I'd mentioned just there, we have our own cohorts that are extensively examined, we've been early and enthusiastic adopters of the UK Biobank data. It's a fantastic resource. I think actually at the last count, me and my team have been involved in more than 100 research papers and projects, either leading or involved in since the Biobank started up. And it's allowed us to ask some really, really interesting questions. I'm going to just pick 3 that I think make quite a coherent narrative for you here. And 3 areas that I think have made really good contributions to. So the first is how does the brain age, so characterising aspects of brain ageing. And there's still work to be done on that. What are brain biomarkers of cognitive differences, and do those change as we age? And what are some of the modifiable factors, and we've touched on that before, so things that we might be able to change that might influence brain ageing. So this first question, how does the brain age? So the first release of UK Biobank data was, I think, either 2015 or 2016. And we recognised instantly with some of the brand new measures that the UK Biobank team had worked hard to include in the brain scans that you took part in. We were able to do some characterisation of these brand new facets, these brand new ways of measuring parts of the white matter connections in your brains. It was, as I say, one of the first papers to use the first Biobank brain imaging release. And these are just some representations of the major pathways that we can measure. So in lots of detail, we can get into even more detail than this now, but 27 major pathways. And these represent thousands and thousands and thousands and thousands of these tiny, tiny nerve fibres that communicate information between these different areas of the brain. And so we derived, first of all, a general measure. So across all of the tracts of our brains, all the different pathways in our brains, to what degree are they changing, or to what degree are they different between younger and older participants in UK Biobank? Now, unfortunately, it looks like some things are going up, which is good, but actually, those things going up denote probably worse things happening. And so, in fact, actually, throughout all of the different measures, these different facets that we were able to measure of the white matter tracts, generally speaking, things are declining, they're getting worse. But also, can you see how they're slightly bendy lines? That shows a degree, perhaps, of acceleration as well. So things are maybe getting increasingly worse. But we also found that much, but not all, of white matter aging can be characterised in this general way. And we find that there are specific and different aging patterns across individual pathways too. So we can look at these here along the bottom are just the individual pathways that I showed you before, just these list of different pathways. And we've just flipped it now so that the lower the dots are on this line, the stronger the negative effects are, so the worse things are getting. And you can see how they're not equal at all. So some of these tracks are showing relatively small aging associations, and other of these tracks are showing much stronger associations. So it's not all equal, and actually you can visualise that much more clearly here. So with warmer colours being much stronger aging associations. So you can see pretty clearly there that it's important to be able to look within the white matter tract. It's not just a global aging process. And those patterns are important for what comes next. So the next thing that we were able to contribute to was brain biomarkers of cognitive function. The first thing that we tackled in this next paper was the very long-running question of is bigger better? So is it true that having a bigger brain is slightly better for your general cognitive function in aging? All this shows you is that we took 4 cognitive assessments that were available at the time, that were sat at the same time as the imaging appointment, and derived a general measure of cognitive function. That just describes the fact that on average people who perform better on one type of test perform better on others. And we asked in, this time, 20,000 people almost, having more brain, is it related to having a higher cognitive score? And we found that it was, but not perfectly. So in other words, it's not true that every single person who had a bigger brain performed better on their cognitive skills, but rather that there was a general tendency across all people for those with a bigger brain to, on average, perform a little bit better. Incidentally, this paper more than doubled the entire world literature on that question. And this is only really a side question to the main thing that we were doing here. But it just gives you another example of the things that we're able to do together. So bigger is a bit better, but fundamentally, understanding just things at the global level isn't all that informative. The brain is a hugely complex structure, as I'm sure you can see And so just asking about how big is it, how much of it do you have, is not really going to get us very far in terms of understanding cognitive differences, cognitive aging, and its relationship with the brain. So then we broke it down a bit further. So we asked, okay, well, is it true that if you have a bit more grey matter, a bit more normal appearing white matter as opposed to the white matter hyperintensity, those white matter scars that I showed you at the beginning, and then two facets of the long-range white matter pathways that I showed you just now, is it true that information about all of those helps you build up more of a picture of people's brain differences? And for the most part, that's true, although these two didn't contribute, these two bits of information about the white matter pathways didn't tell us any more than we could already get at with just those overall kind of brain structural measures of overall size. But when we asked about splitting it between the younger portion of the sample and the older portion of the sample, actually we could double the amount of differences that we could explain between people's cognitive abilities using all of these different all these different facets of the brain. And these two measures of the white matter connectivity actually then provided us with additional information beyond what we already knew. So in other words, these seem to be informative markers of what's going on during the process of aging. And then we can break it down into even more detail. And so here we asked again about the regional differences in terms of cognitive performance, in terms of cognitive function, and their associations with the different facets of our white matter pathways. And what we found was, again, if you plot the associations here, the strength of the links, if you like, between having better health in your white matter connections and better cognitive performance, what we found was actually that those same fibres were coming up as we found for aging. So in other words, unfortunately for us, those fibres that are most strongly linked to our more complex cognitive skills are actually those fibres that are most susceptible, it seems, to aging too. And then finally, again, we've already mentioned a bit about modifiable risk, and we're looking at vascular risk factors, so things like your blood pressure, things like your whether you smoke, your body mass index and your waist-hip ratio, things like that, whether you have hypertension. And what we found was actually that so I said effects on brain health, so what I mean is the associations here, the links between having more vascular risk and having poorer brain health was small, but pervasive. So in other words, across all the types of measures that we have of the brain, there seem to be elevated vascular risk and poorer brain health measures. And again, although some of these vascular risk factors seem to co-occur, so for example, if you have hypertension and a greater waist-hip ratio and BMI, you might also have diabetes. It's not always the case by any stretch, but there seems to be unique information being conveyed by all of these different factors beyond and above others on our brain health associations too. And what we've got here is an overlap of, if you have 1 or 2 or 3 or 4 or 5 of these vascular risk factors, the kind of the yellow and the white colours in particular here show areas where there seem to be overlapping associations. So it seems there's almost a kind of a confluence of effects over specific areas of the outer cortex of the brain. And that's also true for these white matter pathways. So when you dig much deeper into the regional associations, you find again that those brain areas most susceptible to age and most important for our cognitive functioning were also those areas that appear most strongly associated with our vascular risk factors. And so in relation to John's question, we're building up a picture whereby actually there seems to be this subset of connections and subset of areas in the brain that seem to be most important for cognitive skills, most susceptible to aging, and weakly also associated with vascular risk. Of course, there are many other things. This is a relatively small association here, but again, one of the values of UK Biobank is being able to use huge samples to reliably detect these really small effects that otherwise might go unnoticed. So there are many other genetic and lifestyle environmental factors that we're looking at, and of course, work is ongoing to work out how those also relate to our brain structure. So, UK Biobank, a fantastic resource. Having the small effects, being able to be reliably detected is hugely valuable. The brain information that you provide is extremely high quality. Having all of the other data to put together with it is unrivaled. And as well, it's a well-curated, managed and orchestrated data resource, which makes it hugely attractive to researchers like us to go and apply and use those data. And there's, of course, lots of additional work ongoing with us and with many other researchers too. So I want to thank you very much indeed, and also the Biobank Collection and imaging teams, and also my collaborators and funders. So thanks so much for your attention. Cheers.
So yeah, I'm conscious that we are overrunning a little bit, so I'll be as quick as possible. But now you've heard, all this really interesting information and research that's going on with the data, I just thought we'd bring it back to the visit itself. I think the majority of you in this room are participants who've received an invite but haven't yet attended a visit. So I just wanted to try and give you a flavour really of what it's like to take part. So a very, very brief background. So we are looking to image 100,000 of our original 500,000 participants, and that involves having an MRI scan of the brain, of the heart, and of the abdomen, basically your whole body from your shoulders down to your knees, a DEXA scan showing the bone density of your bones, and an ultrasound of your carotid artery at the side of the neck there, looking at the thickness of the artery wall. We also do a 12-lead ECG, looking for arrhythmias of the heart, and we will offer at least a cardiac monitor for you to wear and take away, and you wear it for another 2 weeks and then post it back to us. And then there is a repeat of all of the measures that you would have done as part of the baseline study. We've also just started doing repeat imaging, so that's where we're looking for 60,000 participants from the original 100,000 to come back and do it again in a few years' time, and that helps us with some of this longitudinal data that you've heard about today. Very brief timeline of how we've got to where we are. So the funding for this project was actually agreed in 2012, and we began a pilot study in Stockport in April 2014. That pilot was confirmed as being successful some 2 years later, and that released the funding to continue that baseline study, but also to open up the other sites. We opened our site in Newcastle in April 2017. We also did a short repeat study called DP UK, which is specifically around here, the dementia platform UK study. We opened a Reading site in 2018. Our Bristol site opened perfectly February 2020. It was open for 4 weeks before we closed again. And I will quickly mention that. So all of the sites closed in March 2020 for nearly a year. But we actually opened up again to do that SARS-CoV study that Paul spoke about for some 7 or 8 months. We had 2,000 participants come back for a repeat study, and that in itself has led to some really, really interesting research. That was a true, unique project across the world. We were the only study able to do anything like that. There's lots of studies looking at the brain effects after COVID, but nobody else had the images from before COVID as well. So really, really important. And that gets us basically to today, where the repeat imaging in the 60,000 has now been funded and is starting. And that has begun up in Newcastle, where the baseline element is drawing to a close. We're just in the last sort of 1,000 participants really who will be going to Newcastle for the imaging up there. So the centre nearest to Leeds, and I realise near is a very general term here, but it's in Stockport, so just south of Manchester. And we only have the 4 sites basically because these MRI scanners are very expensive and they are very difficult to move. So unlike the original assessment, where we basically opened up a small clinic in the towns and cities around the country, ran them for 6 months, As soon as everybody from that small area had attended, we closed it down and moved to the next town along. We just can't do that for the MRI scanners. So we have the 4 sites, and they basically invite participants from all the assessment centres for about a 50-mile radius. So the Stockport site will obviously have the Manchester and North Manchester participants attend, Sheffield participants, Nottingham, Stoke and Birmingham will get invited to Stockport, Chester and North Wales, Liverpool, and right up to sort of Cumbria and Leeds. So if you receive an invitation, then your nearest centre is Stockport. That doesn't mean you have to go to Stockport. If you have relatives up in Newcastle, or you think the train up to Newcastle is easier, then that's fine. You can make an appointment at the other sites, but your invite will be for Stockport. The Stockport Centre itself is just here. So this is the M60 Manchester Ring Road. So I made the trip this morning here. It was a fraction over an hour, and that was in relatively poor driving conditions. So it's not too far away. We're also close to the train station in Stockport, and we have a taxi service that we put on to get participants to and from the train station if you decide to catch the train. The site itself, and this goes for all of the sites, it's built in a warehouse. So we're not in a hospital, we're not a clinical facility at all. It's in a warehouse on the outskirts of Stockport, and that is done for a couple of very good reasons. One, it was relatively cheap to hire a warehouse. It meant you can get around all the administrative pain of having to be registered at a particular hospital to go to their site. It meant that you could literally roll the MRI scanners through the big shutter doors and then build a room around them. So this one, like I say, it's about a mile from the train station. It's not too difficult to find, but some participants do get a little bit wary when you start going into an industrial estate. So just to give you an indication here, you turn left, carry on down Europa Way and turn left into Spectrum Way, and there's our centre again, a beautiful little warehouse there. And the large white tanks you see, that's actually the liquid helium that's used to, liquid nitrogen, that's used to keep the samples frozen. So they are also stored in this warehouse. So all the blood samples that you gave during the baseline assessment are in a freezer here. Inside the site, you get a very normal sort of reception area where you will come in and register at the front desk, and you have to provide your consent again to take part in the imaging study, and that consent includes allowing us to share any incidental findings that might appear with your GP. And to very briefly touch on that, so this is not a health visit, it's not a health check. We are not looking for any sort of illness. If we do see something, so obviously when you're taking a brain MRI scan, you have to look at the brain in order to collect those images. If we see something, then we would write to your GP with details of what that is, and we'd write to yourselves and ask you to get in touch with your GP. But if we don't write to you, and you've been having terrible headaches for several weeks, don't think, oh, everything's okay, I've just had my biobank visit. That is not the purpose, okay? So you do your consent, and then you come through and you will have an MRI scan. Now this is one of our MRI scanners. You can see it's quite large. The bore, the hole that you go through, is a wide bore. It's bigger than most clinical MRI scans, so hopefully it won't feel as claustrophobic as you might fear. That's the view that we get from the desk where we operate the scanner from. This is a DEXA. A number of you may have had one already, but it's basically you lie on the bed and we will position you in a number of ways on your back and on your side, and the arm there will travel down the bed and back up and over you, and it gives you a little x-ray. In terms of the images themselves, this is a sample from the MRI of the body that we will collect. We'll get a nice functional scan of your heart beating. These are the functional brain MRI taken during the task, again highlighting areas of activity during the tasks. And this is the scan of your ultrasound, again showing the wall there of your carotid artery. And this is the sort of image that we get from the DEXA. The image itself, as I said, you arrive, you have the welcome and the screening, and then you have your brain MRI scan. And the brain MRI is about 40 minutes long. I'll aware here, the slide is supposed to make sound here, but it's not connected up to the speaker, so I'm just going to attempt this through my mobile phone. Apologies if this doesn't quite work out, but we'll see where we get to. I think you get the point. So, very noisy, but absolutely normal and absolutely safe. So when you get on the scanner, you'll be given earplugs to wear, and they do keep out a lot of the sound, but you can still hear it. And as the magnets themselves turn on and off very quickly to help generate the images, it can feel weird. The bed sometimes vibrates. It can be unnerving, I'm not going to lie, but it is absolutely normal, and millions of these are done every year without any sort of incident, so please don't worry. So after 40 minutes of that sort of noise, you have the heart and abdominal scan with similar noise, and then you'll have the DEXA and the carotid with very little noise. You'll be happy to. Once you've done that, then we'll collect some samples from you again, and you do that repeated baseline before you leave. And that entire visit is roughly 4.5 hours. Clearly, it does differ from person to person, but it's significantly longer than your baseline visit, which would have probably been about an hour or so. But we get an awful lot of data through that visit. The visit itself, so we are open all day. So our staff get on site at 7:30 in the morning, and we close at 8:30 in the evening. Our first participant appointment is at 8 a.m. And generally we have 3 participants turn up at the same time, go through that registration consent screening, get changed out of their clothes into MR-safe clothing. And then you will go into one of the 3 scans. And when the first scan is done, you swap, and so all 3 people go along in parallel. And when you come out of the imaging and do the baselines, the next 3 participants are ready to go into the scanner. So those scanners are worked from, the first person gets on the brain scanner at about 8:20 in the morning, and they will get off at just before 8 o'clock at night. So we are as quick and efficient as we possibly can, and in terms of trying to get most people through in the time that we have available. So we do 18 participants per day. We do that 7 days per week, and we're open not quite 365 days per year. We close during the Christmas break, we close over Easter, and we close for the odd maintenance day, but pretty much we're open all year round. So that's led us to today, where we've already scanned over 65,000 participants, and because Stockport opened that much earlier for the pilot, they've done significantly more. So we've done 35,000 of those scans in Stockport. We've also done 3,000 repeats for the DP UK study and the 2,000 COVID studies that I spoke about before. Baseline imaging in Stockport and Newcastle, for that matter, is drawing to a close. So we think we are probably open in Stockport for another 6 months or so. So if you are interested in coming along, then now is the opportunity. The sites in Reading and Bristol will still be open for baseline participants for another year or so, but clearly they're a bit harder to get to. And yeah, the repeat imaging has started. So we hope that you'll join us soon.